Welcome to another important topic in the genetics, that is independent assortment. This is an inheritance pattern of genes present in the parental organisms to the next generation. Independent assortment. Independent means freedom or freely. Assortment means separation. The genes are alleles located on the homologous chromosomes in the parent are separated freely during the meiotic division and involved in the crossing over, form the new genetic recombinations, entered into the gametes, form the new genotypes or phenotypes in the next generation. In this class, I'll tell you one principle, one example, and give you the complete concept. After the complete discussion, if you remember this principle, if you understand that example, you can take one question from any examination, you can keep it in the principle, you can get the answer. Then what is the principle of independent assortment? Two or more genes that determine different characters located on the same chromosome or different homologous chromosomes separated independently during the meiotic division and enter into the different gametes. It means whatever it may be the allelic pattern present in the parental organism, either it may be homozygous dominant or it may be homozygous recessive or heterozygous dominant or other heterozygotes. Whatever it may be the genetical pattern of parental chromosomes, they can be separated, involved in the crossing over, exchanged the genetic material, form the new type of genetic recombinations, entered into the gametes and form the new genotypes and phenotypes in the next generation. That is independent assortment. That's why Mendel got 16 genetic recombinations in the F2 generation of dihybrid. Means Mendel has chosen two homozygous parents. One is the double dominant parent, another one is the double recessive parent. But from these parents, in the F2 generation, he got 16 genetic recombinations. How do these different 16 genetic recombinations are formed from these two homologous parents? That is possible if the chromosomal material is exchanged in between the chromosomes or chromatids during the meiotic division. Does all the organisms follow this independent assortment to transmit the genes or alleles from the parents to next generation? No. There are another type of hypotheses or laws or theories are also there. This means the independent assortment is a way of transmission. Then immediately we can get one question is, how do genes located on the parental chromosomes can be inherited to the next generation? Actually, Mendel has given the answer for this question by the independent assortment, and Bateson and Punnett gave the answer with their coupling and repulsion hypothesis. But the T.H. Morgan with his linkage or chromosomal theory of linkage, the answer is different for this question. So actually for this question there is no only one answer. There are actually three answers are there. So how do the genes present in the parental organism could be transferred to the next generation is by the independent assortment. Means in some organisms on some chromosomes, some of the genes are following this independent assortment and transferred to next generation. But in some of the genes or alleles are following this coupling and repulsion hypothesis, and some genes or alleles located on the parental chromosomes could be transferred to the next generation by linkage. So there are three answers for this question. But in this class I am going to discuss about the independent assortment only. I am not discussing about this coupling and repulsion hypothesis and linkage. This coupling and repulsion hypothesis and linkage, there are two other videos are also there. The link is given in the description box. You can go through those videos compulsory after completing this video. So now I am concentrating on this independent assortment. Okay, in this independent assortment, the genes or alleles located on the chromosomes of sweet pea plants, Pisum sativum, assorted independently or separated independently during the meiotic division and enter into the different gametes by involving in the crossing over. That's why they formed new type of genetic recombinations in the gametes as well as in the next generations. In this independent assortment, Mendel has conducted many experiments on the pea plant, Pisum sativum. He has taken different parents. Sometimes he chose homozygous dominant parent, homozygous recessive parents, and sometimes heterozygous dominant parent with the double recessive parent, and other type of many experiments were conducted by the Mendel and got the results. Those results are accumulated, statistically analyzed, and finally framed this independent assortment. Mendel has observed 7 contrasting characters in the pea plant. Those are round seeds, yellow pod color seeds, white flowers, full pod and yellow pod, axial flowering and long stem. These are the dominant characters. Contrasting to these characters, there are 7 recessive characters are also there. These are from wrinkle, green, violet, constricted between the seeds, green color, pod color, terminal flowering and the short stem. These are the 7 type of characters. At a time, Mendel has chosen 2 parents. Those 2 parents are homozygous parents. He continued the dihybrid cross or dihybrid hybridization and got the 16 genetic recombinations in every experiment, every dihybrid cross. With this, he proved that in the dihybrid hybridization or dihybrid cross, the genes or alleles present in the parental organism are separated during the meiotic division and involved in the crossing over, formed the new genetic recombinations, entered into the gametes, and the next generation is with different phenotypes. Those 4 types of phenotypes, 9:3:3:1, and the different genotypes are formed. Then how did these different genotypes, different phenotypes are formed from this only 2 types of homozygous parents? How is it possible? Because these parents are involving in the crossing over, forming the new genetic recombinations, so they can able to form these different genotypes and different phenotypes in the next generation. This is the proof of independent assortment. If there is no crossing over, only parental type organisms are formed in the next generation. This is the example I am going to take from the experiments of Mendel. That is, this is a homozygous dominant parent, yellow color seed and the round seed. This is the double recessive parent, green color and wrinkle seeds. So allowed them for the cross fertilization. Gametes are formed during the meiotic division. Same types of gametes are formed from this parent as well as from this parent also, because these are homozygous parents. They can able to form only one type of gametes. In the F1 generation, offsprings are formed. These offsprings are containing the heterozygosity. Dominant genes are located on one chromosome, recessive genes are located on another chromosome. This is F1 heterozygous dominant progeny, because it is these recessive characters suppressing by these dominant alleles. At this time, Mendel got one question is, where are the recessive characters, green and wrinkle seeds? Whether the recessive characters are present or not, whether they are hidden or they are gone away from this population, he has to prove. So he has taken F1 generation plants, male and female plants, allowed them for the self-fertilization. This is the F1 male, this is F1 female. Both are heterozygous dominant parents. From this parent, how many types of gametes are formed? This is the gamete formed from this. And this is the gamete, same, formed from this, which is containing the recessive alleles only. But if the crossing over take place here or here, they can form these 2 gametes also, genetically recombined gametes. So means from this F1 parent, 4 types of gametes are formed. From this parent also 4 types of gametes are formed, same gametes. So From one parent, the gametes are arranged on the checkerboard this side. From this parent, those gametes are written on this side. So the genotypes are formed. 16 genotypes are formed with 4 phenotypes. Here actually yellow round is the dominant character. Both are dominant characters. Yellow wrinkled. Yellow is the dominant one, wrinkled is the recessive one. These two are appearing in this phenotype. This is green round. Green is the recessive character, round is the dominant character, but both are appearing in this population. Then final one is the green wrinkled. This is an important observation. This is double recessive progeny. This double recessive progeny, this is the double recessive progeny. This progeny is formed in the F2 generation. Recessive characters are hidden in the F1 generation, but those are reappearing in the F2 generation. Means during the meiotic division, the alleles or genes present in the parental organism are exchanging the genetic material in between the non-sister chromatids or chromosomes. That's why they formed different types of gametes. In these different types of gametes, you can see these two are the dominant alleles. These two are the recessive alleles. In these two alleles, one is the recessive allele and another one is the dominant allele. As well as and this chromosome also, one is dominant allele, another one is the recessive allele. How did these recombined gametes are formed? Because of the crossing over. So ultimately we got the answer is the genes or alleles present in the parental organism are involving in the crossing over, forming the new genetic recombinations while they are forming the gametes, entered into the next generation and form the new genotypes and new phenotypes. That is proved the independent assortment. Then test cross. What is test cross means? The F1 dihybrid parent is cross fertilized with a double recessive parent. Double recessive parent is the green and wrinkled. This is called the test cross. What is testing here? This F1 parent is heterozygous dominant parent. If it is the heterozygous dominant parent, what is its genotype? That is to be tested. So this F1 dihybrid is test crossed with the double recessive parent. Double recessive parent is very important to know the allelic pattern of F1 parent. Here you can see what are the gametes produced from this F1 dihybrid parent. This gamete is same. This small y, smaller gamete is also same. These two gametes you have to observe. If the crossing over take place here or here, they can form these two types of gametes. These two types of gametes are different. Okay, these two are genetically recombined gametes. So each gamete is fertilizing with the other gamete, formed these 4 phenotypes. These 4 phenotypes are yellow round, this is dominant parental like organism. Green round, this is recombined because green is the recessive character, round is the dominant character. This is yellow wrinkled, this is also recombined progeny because yellow is the dominant, wrinkled is the recessive. Green wrinkled, this is double recessive parent just like the homozygous recessive parent. So homozygous recessive parent formed, homozygous dominant parent is also formed, double dominant, and 2 recombinants are also formed. Actually all these are formed in 1:1:1:1 ratio, that is equal ratio. That means the genes or alleles present in the parental organism are involved in the crossing over, separated during the meiotic division, entered into the different gametes with new genetic recombinations and formed these 4 types of phenotypes in equal quantities. This test cross is proving 2. One is to know the exact genotype of F1 parent and also proving once again the independent assortment. Finally, comparison of independent assortment with the coupling repulsion hypothesis and the linkage. In the independent assortment, new genetic recombinations are forming with the crossing over. But whereas in the coupling and repulsion hypothesis as well as in the linkage, no new genetic recombinations. They can form only parental progenies because there is no crossing over. So why I am comparing these three theories, we can get one question. Which one is the correct inheritance pattern of genes? Independent assortment or these two? Which one is correct? Which one has to be followed? Which one is considering? All are accepted theories and considering now to preparing the gene maps, linkage maps, know the inheritance pattern of deceased alleles or genes, and every application in the genetics. So this means some genes or alleles located on some chromosome in some organisms are following this independent assortment. Some of the genes or alleles are following this coupling and repulsion hypothesis, and some are following the linkage. So everything is taken into consideration. And another important question from this discussion is, which theory or hypothesis 100%? Is there any 100% proving theory? No one is 100%, because in the independent assortment also some parental progenies are formed. You can see them here. Is it the parental progeny or not? Is it the parental progeny or not? Wait. This is the parental progeny, the dihybrid cross. This is also parental progeny. Of course, these are the genetically recombined, but parental progenies are also forming. So independent assortment is also not 100% independent separation. So coupling repulsion hypothesis also containing some genetic recombinations, as well as linkage also form new type of genotypes in the next generation. Why they were proposed this independent assortment, this Bateson and Punnett proposed this coupling and repulsion hypothesis. Why the linkage is framed? Because from the experiments of different scientists, they were choose the majority of the population. So majority of the population are containing the different inheritance pattern. So by taking those conclusions, these theories are formed. So means majority of the population in the independent assortment are genetically recombined. Majority of the population from the coupling and repulsion hypothesis are no new genetic recombinations, as well as in the linkage also. So this is the comparison. These are the differences